Station, this is Houston. Are you ready for the event?
Houston, this is Station. We are ready for the event.
National Institutes of Health, this is Mission Control Houston. Please call Station for a voice check.
Station, this is Dr. Francis Collins with National Institutes of Health. How do you hear me?
Hello, Dr. Collins and NIH. I hear you loud and clear. Welcome again to the International Space Station.
This is such a rush for me, Astro Kate, being able to have another conversation with you 5 years later, but you're up there again. And you're doing a lot of interesting science that we want to get into. I just want to say thank you for what you are doing to push back the frontiers of genetics and molecular biology in this remarkable environment that you're in the middle of right there. And I also have to say thank you for coming to NIH for our DNA Day a while back. So this is our third chance of being able to talk with each other. And you are a hero to all of us who work in molecular biology and biomedical research for what you're doing there. And what a role model you are for so many who are maybe thinking about going into science or dreaming about what it would be like to do science in space. And you're not dreaming it. Well, I suppose you dream it sometimes too, but you're doing it. So thank you for all of that.
Absolutely. And for folks that are thinking about doing science in space, I would say it was a dream of mine at one point. So keep dreaming and keep doing. It's pretty amazing up here.
Well, last time we talked, I told you that by doing this conversation, I'm getting super credibility with my 5 grandkids. And that's being repeated here today, especially because of COVID-19, they're virtually going to school. So maybe they're even playing hooky and pretending to be on a Zoom call where they're listening to us. I wouldn't imagine anyone would ever do that. So yeah, thanks for giving me that kind of visibility. And oh my gosh, Kate, I did had somebody tipped off to me that you had a conversation with the director of the National Science Foundation. And somewhere in there, you got asked about your favorite scientist, and I was pretty blown away by your answer. So I don't know where to begin. Talk about getting credibility for my grandkids. That was over the top.
Oh, that's awesome. I get called a hero sometimes, or people look up to me, and it's always kind of a little bit uncomfortable and embarrassing because I'm just me and I'm just doing what I love. So I hope I did not embarrass you too much. But it was an honest answer to a great question.
Yeah, I'm just me and I'm doing what I love too. Well, let's talk about it. So you're up there this time. You've been there since like October. So this is going on 6 months, and must have been a lot of experiments that you had to do to stay up there all that time. Can you tell me something about what's happening with the tissue chips? Because that's a collaboration with NIH that we're very excited about, having these chips that have human cells on them growing in a way that we can assess what the effects are of gravity. But tell me about the experiments you're doing this trip.
Yeah, absolutely. The tissue chips are really phenomenal, and I've been watching this program for a while. We say tissue chips, and we sometimes will kind of think of things like silicon chips. They're not necessarily silicon chips, but they're small, modular cell culture system where really the key thing is that we can study tissue in what's kind of a natural environment and tissue types that more replicate what's going on in human organs. So, for example, we had a cardiomyocyte experiment when I was up here in 2016, and that was a really cool experiment, but we were just looking at cardiomyocytes by themselves. These are the main heart cells. What we can do with the tissue chips is actually then add fibroblasts and endothelial cells. So we start to replicate more of what it's like to see real tissue like you would see in a human body. These are super exciting to understand the effects of spaceflight on the human body, but it's not just all about NASA. One of the things that we're looking at with these tissue chips is how we can replicate some disease processes on Earth. Like, for example, cardiovascular disease. We've got a lot of parallels for what happens to humans in microgravity that look like processes that are dysfunctional in cardiovascular disease or in aging. We get this really rapid acceleration of a lot of these processes. So I'm a pretty healthy 42-year-old human. I did launch in October on my birthday, but some of the physiologic aspects that we see of human biology and astronauts look like disease and aging on Earth. So the tissue chip study is allowing us to take small pieces of those kinds of tissues. Another one we looked at was bone and synovial fluid, looking at osteoarthritis. And then we can study microgravity actually as a variable in these systems. So there's very, very exciting for space science, and I think a really cool partnership between NASA and NIH.
Yeah, that sounds highly relevant, and I'm glad you pointed out it's not just about what we need to learn about health for astronauts, including those that may go to Mars someday and maybe in microgravity for a long time, but also it's sort of a model of the normal aging process, but maybe accelerated. Biomedical research is always looking for good models of common disorders that may be easier to study in the model system. And what do we know at this point? I mean, I know there's a lot of concern about bone loss. I'm sure that's something that you must be thinking about, being up there for months at a time, just because we know that somehow gravity seems to help with strength of bones. That's why I did my weightlifting this morning, because it was supposed to help keep me from ending up with fragile bones. But have we figured anything out yet about how that works? How does something like gravity, which seems like a cell would hardly notice, how does that actually have an effect on its biology? Are we still on that path of trying to sort that out?
Yeah, absolutely. And you don't think of gravity as being, the physicists don't call it a strong force. I didn't notice it until I came back to the planet last time. And then, oh boy, did I really notice it. It affects everything. And so just the fact that humans have weight on the planet, and we walk around and we impact, all the time. When you're taking a step, you're getting impact and you're getting loading on that bone. And we've evolved like this, and it's constantly sending signals to our bone to regenerate. So we think that one of the things that happens in space is we're floating. I've got no impact on my bone here because I'm just talking to you, and I float in any direction. I'm not walking. I'm not stepping. So if you lose that signal all of the time, it's use it or lose it with the human body. And so the body says, I don't really need that much. I'm going to spend calories in my brain or somewhere else. And so we start to lose bone. The problem is when we lose bone, when we can see this with younger astronauts, we go back to gravity and we can gain back bone again, but we gain it all around the cortex. So your bone's like a PVC pipe, and you'll gain a lot of the bone density back again. So it's not that problem with bone density loss, but you lose the trabecular structure. So this mesh that's in the middle of your bones that really contributes to the strength, your ability of your bones to not break. Once you start to lose that, even if you're young and healthy like an astronaut, you can't grow that back again. So it's a very interesting model for osteoporosis. And one of our biggest countermeasures is exactly what you did this morning, which is what I did this morning too, which is lift weights and lift really heavy weights. That helps load up your bones, and that can prevent ever losing that trabecular structure.
So everybody listening aren't quite sure about bone anatomy. After this, go and look up the bone cortex and the trabecular structures, and you'll see what Kate is talking about there. Very interesting. You might think bones are just really simple. They're really quite elegant and complex, especially when you get down to the individual cell layer. Well, I want to hear also about where you are with DNA sequencing. After all, Astro Kate here was the very first person to sequence DNA in space back 5 years ago using a nanopore sequencer. And I think I heard you sequence something like 2 billion base pairs while you were up there with this instrument floating around, sequencing viruses and bacteria. But I gather it's gotten even more sophisticated in this trip. I think I saw a little clip of you doing something that looked like multiplexing. And I have to ask, before you tell me about the exact nature of the experiment, how do you do experiments in microgravity where you have tubes and you have pipette men and you don't want everything to sort of float around and get away from you. Is there like stick 'em somewhere that you put everything down so that it doesn't sort of disappear or float away?
Yeah, it's actually one of the hardest parts about doing laboratory science here is that you can have a bench, but nothing will stay on the bench. It moves around and it walks away and it floats off the second you stop looking at it. So we it's very low tech. We use a lot of Velcro and we use a lot of gray tape up here for sensitive electronics. We use Kapton tape, so literally tape and Velcro. But these are some of the challenges of doing experiments in microgravity. In 2016, we really started doing a lot more with pipettes. It wasn't something that we've done a lot up here. It's hard to manage. Instead of having a pipette holder, I just put a big piece of gray tape and turned the pipettes upside down and kind of stuck them on the tape. And they sort of wave around a little bit, but they're held in place enough by the stickiness of the tape. One of the things that I just solved, like in October, November, when I got up here, is what to do with the pipette tips. So normally you just put your pipette tips in a bucket on your bench. Can't do that up here. They float right back out again. So we were taking them off individually and putting them in a plastic bag. And that was not very efficient operation. So I figured out how we can use a Sharps container with most of the Sharps container closed. And then that actually keeps it. It's literally stuff like this that I think is advancing molecular biology up here because it's such a unique and weird environment when we're all in free fall.
Okay, that's cool to try to imagine that. But now tell me the experiments you're doing with DNA sequencing this time. Looks pretty complicated.
Yeah, so you're right. We were doing some multiplexing, and that multiplexing in itself, it's not a huge advance. We do this all the time on the ground for large-scale sequencing experiments, but it is pretty cool. So sometimes you'll hear this referred to as DNA barcoding. So you can take a little piece of DNA that's an individual barcode, and you can anneal it to whatever you're sequencing, and then you sequence through all of that. And what that lets you do is take a whole bunch of different samples. So I could prepare 96 different samples, each in their individual well, and then mix them all together and just do one sequencing reaction. And then you can read those little barcodes and deconvolute back out that data. So we're trying that up here because one of the interesting, we've got lots of constraints that you don't have on the ground. Things like power and data are really constrained in this spacecraft environment, but crew time is really constrained. And so we don't want to run 96 different sequencing reactions if we can make this more efficient. So we haven't gotten to 96 yet. We started at, the ground started at 3, and I was like, I've got to strip 8 here. Let's do 8. So we did 8, but this is pretty scalable to 96. I don't think that's much of a problem. We are experimenting with multi-channel pipettes as well, and that seems to be working really well in space, surprisingly enough. And so I think we're moving to an era of much higher throughput in terms of our sequencing up here. We are still using the nanopore technology because the larger sequencing machines are just not super suitable for the space environment. We like the small portable machines because they don't have lasers that you have to adjust or lots of power requirements or huge space requirements. So the kinds of things that you would think about for a bench-to-bedside type real point of care, something that you might want to have in the clinic for sequencing someday, or things that you would do in remote studies and field work. I did a lot of work in Congo, and something like the nanopore sequencer would have been super helpful out there where you're very removed. The space station is definitely a field laboratory.
Yeah, the nanopore sequencer is almost like the size of a cell phone, so you don't have to have a big bench space to place it on. Yeah, people who may not know, Astro Kate, before she was an astronaut, was a researcher, PhD from Stanford, I might say, and was also a researcher at the Whitehead Institute and did remarkable research on things like Ebola and Lassa fever in doing field work in the DRC. So, yeah, she knows about field work, that's for sure, in lots of different kinds of areas. So what are you sequencing now? What's going into these 96 wells when you decide you're going to multiplex them?
Yeah, so this is the original driver for most of the sequencing development work that happened was, we are in a microbial environment up here. We've tracked it for a long time by taking samples and putting them on a culture plate. But then we have to wait for a space vehicle to go back to Earth. And sometimes this can happen 3 months later. And so we only get the sample right before the space vehicle returns. We can take culture plates up here and we can kind of grossly identify them, but we don't know what's growing. And we don't love taking everything through a culture-based method if you're going to really do environmental metagenomics. So sequencing is kind of this technology we want to develop to be able to swab the wall and say what's growing and get that answer back right now and not have to wait for a spacecraft to come back to Earth. So one of the things that we've multiplexed is I went and found the richest microbial locations on the space station. For your grandkids that are watching, these are the grossest places I could possibly find, and I swabbed them, and that's what we multiplexed. We've got another study that's not doing direct sequencing right now, but that is, it was about 800 different swabs. This one took me like a little over a week, 2 weeks, and we're doing a full spatial profiling of the International Space Station with about 800 different swabs and all these different modules. We'll sequence those back on the ground, but these are some pretty cool and complex studies.
Wow. And so that will pick up both bacteria and viruses and fungi, all kinds of living things that presumably are there because the people are there. They didn't spontaneously arise.
Yeah, so we've got a really rich microbiome up here. We think, wherever humans go, they bring a microbiome. But one of our questions about spaceflight is because this is a completely artificial environment, there's no dirt up here. There's no interaction with the outside at any point. Anything that comes up to this environment is launched from the ground. And oftentimes it's not necessarily sterile, but it's packaged in a clean room, and microbial counts are attempted to be reduced. So one of our questions is, what does that do? Are we selecting for certain microbes? Are there microbes that grow better in the space environment because of microgravity or radiation? And is this a bad idea? Humans may need microbes. Do we have a healthy microbiome up here? We probably don't have a lot of pathogenic bacteria, but is our natural environment one that we want to keep having up here when we have spacecraft and really long duration times in a spacecraft?
Which is coming one day not too far off. Certainly for kids who are listening, you're going to be watching that first flight to Mars at some point. Which brings me to my last question, Kate, which is, okay, DNA sequencing applied to organisms from planet Earth. We think we know how to do that. We know that all living organisms have DNA or RNA. But if we're looking for life on Mars, as the Perseverance Mars rover is right now on this trip, what do we think we would find? How would we know it? Would we have to run your DNA sequencer before we were convinced that it was life and not just something else? And wait a minute, why should life on Mars have DNA anyway? What do you think about all that?
That is a great question. These are some really good astrobiology debates and discussions, and I don't know that it's going to be a strict DNA type signature. So I think that sequencing is going to be one of the modalities that we use. It's one of the tools in our toolkit, but we'll probably use quite a bit of other things. We may be looking for evidence of things like protein, things that can catalyze a reaction, metabolic signatures. Certainly for the sequencer, if it's a sequencing technology that can take base pairs that are chemically a little bit different and handle it, and that's one of the things that the nanopore sequencer can do, it might be better to be a little bit less constrained with knowing what your inputs are. Certainly portability is good, and low power draw. The Perseverance rover doesn't have technology to sequence on it. That's really working on sample return, and that's going to be a huge, long process to get samples back from Mars. But I think our next generation of robotic and human exploration is going to be very, very interested in this question. Sometimes people actually they bypass the moon and they go right to Mars, but I'm very interested in the lunar surface as a negative control for what we're going to find when we go to Mars. When we take humans to Mars, they're going to have a lot of microbes with them. We're going to try to contain this as much as we can and reduce the amount of biological material. But what a great way to have a library of all the things that come with us and our spacecraft on the lunar surface, which we know has no microbes on it.
Yeah, absolutely. Great points. Well, Dr. Kate Rubins, it is an absolute privilege to be able to talk to you. I know you got to talk to the Vice President of the United States 2 days ago, so I'm feeling like, oh my gosh, is she going to even want to talk to me anymore? But this has been a lot of fun. And again, you're doing great work up there, and we're all fascinated by the way in which science is being conducted right there on the International Space Station. And I hope people are watching this, dreaming a little bit themselves, could imagine doing something like this. It's a path forward. This is the best time, tell me if you agree, to be a scientist in the history of the planet, because so many things are possible, even on planet Earth and even in the space station. So thank you for being a wonderful role model for that kind of amazing, exciting life.
Absolutely. And I have an extremely exciting C. elegans experiment that I took a break from the microscopy to do this discussion with you. So this is, not only is this an exciting general time in the space station history, I'm having fun actually today, right now. There's a lot going on up here, and I really do think that science is an incredible career, whether you're doing it in space or on the ground. If people are interested in this, the pleasure and the gratification of being a scientist every day, it's truly amazing to get up and discover new things.
Oh, that's great. And C. elegans, no less. Okay, everybody go look up the roundworm C. elegans, first organism to have its genome completely sequenced that was made up of multiple cells. So there we go. It's all connected. And yeah, I guess you're probably checking out their muscle function too, given that it's a gravity question. So yeah, go to it. Let me not interrupt your further explorations and discoveries. Great to see you, Kate.
It was truly wonderful to talk to you. I'm glad we could welcome you back to the International Space Station, and just incredibly exciting to talk to National Institutes of Health. Hopefully we'll see you post-flight as well.
Station, this is Houston ACR. That concludes the event.
Thank you to all the participants from the National Institutes of Health. Station, we are now resuming operational audio communications.